All right, folks, welcome. We're going to get started here. There'll be a few more people joining the program while we're in transit, but we'll just get started to maximize everyone's time. Good evening, everyone, and welcome to this evening's program and another installment in the Alumni Continuing Education Series, also known as A.C.E.S. My name is Rory O'Neill. I'm the Executive Director of Alumni Engagement here at John Carroll University, and I'm a proud member of JCU's graduating class of 2006. We are so happy you've all joined us tonight for this conversation. The Alumni Continuing Education Series was established in the spring of 2013 by the Office of Alumni Relations here at John Carroll, and it's been established because John Carroll University produces lifelong learners who are eager to stay engaged with their alma mater. These programs have traditionally provided enrichment opportunities for alumni and friends through expertise that's provided by our administration, John Carroll administration, faculty, our staff, and members of the John Carroll alumni community, which we're here to celebrate tonight. For those of you who have not been on John Carroll's campus recently or not been engaged with the university, I just want to provide a few quick updates. One thing to know is John Carroll is on the move. We've opened two new state-of-the-art nursing labs recently to complement our new nursing program, which is our new academic program. We've completed renovations to the DJ Lombardo Student Center and also renovations to Dolan and Bocelli Hall. We recently broke ground on the new Athletic Wellness and Event Center, which will go on top of the Belvoir parking lot. We can see steel being put in every day and progress being made. And this Wellness Center will be a huge addition to our athletic teams and every single student on John Carroll's campus. We are in the middle of a multi-year and multi-floor renovation of Griselli Library. And just in January, we announced that we will be joining the North Coast Athletic Conference in 2025, joining universities such as Denison, Kenyon, and Oberlin, who have national brands and national recognition. We're also thrilled to share that in December, we announced the creation of a College of Health at John Carroll, which will blend the social context and global perspective of a Jesuit liberal arts education and the strategic approach of a business education from the Bowler College of Business. And this will help us train future healthcare leaders going forward. And just as there are exciting developments happening here on campus, there's always exciting developments happening in the John Carroll Alumni Association. That's why we're here tonight, to recognize one of those people, and Dr. Phil Bevilacqua. So before we get started this evening, I'd like to just share a little bit of background as to why we're hosting tonight's program with Dr. Bevilacqua, who's a proud member of the class of 1987. Every member of the John Carroll alumni community experienced the COVID-19 pandemic, and many Carroll alumni worked at the front lines of hospitals, schools, and communities, helping to serve one another in the John Carroll tradition. Our alumni have worked in intensive care units as nurses and doctors, who've worked in schools as teachers and principals, and many alumni kept vital businesses and community organizations afloat during unprecedented times. Carroll alumni have repurposed breweries to make hand sanitizer, and we know of one instance where a decades-old business that sold window drapes was turned around on a dime to start making masks and other supplies to help communities address challenges brought on by the pandemic. The arrival of the COVID-19 vaccine was a highly anticipated and a significant moment during the pandemic. The 2023 Nobel Prize for Medicine was awarded to two University of Pennsylvania scientists named Dr. Katie Carrico and Dr. Drew Weissman, whose research on messenger RNA helped enable mRNA to be used in a drug or a vaccine. When Carrico and Weissman were asked to list the three most influential pieces of research that guided their work, Dr. Bevilacqua's previous research on RNA modification was recognized as fundamental to their work. Our program tonight is a conversation with Dr. Bevilacqua about his JCU education, his career, his research, and his contributions to this year's Nobel Prize in Medicine. We are proud to feature Dr. Bevilacqua, and we hope you all enjoy tonight's program. So just before we get going, a few housekeeping items. Please, we encourage you to view this program in full screen mode. It'll help you enjoy it the best. Tonight's program is a webinar, so none of you will be able to kind of be visible on screen. So please be comfy, get comfortable where you are. Following the moderated conversation, we will have an opportunity for questions and answers from both the moderator and our evening's guest. You may ask those questions by utilizing the chat function at the bottom here, and we will curate those questions during the program, and then we'll ask them to Dr. Bevilacqua and Mike here at the end. Okay. So before we go, I want to introduce our moderator. His name is Dr. Mike Martin. Dr. Martin is a professor of biology here at John Carroll, and he previously served as the Associate Dean of Sciences, Mathematics, and Health within the College of Arts and Sciences here at John Carroll. He has been at John Carroll for over 20 years, and he teaches courses in Introductory to Biology, Genetics, Biotechnology, Molecular Cell Biology, and he's actively involved here in acquiring scholarship grants and support for first-generation low-income and underrepresented minorities in the STEM and health fields. Right, Mike, thank you so much for being our moderator tonight. And I want to, before we hand it off to Mike, I want to introduce our featured guest, Dr. Phil Bevilacqua from the class of '87. He is the Distinguished Professor of Chemistry and Biochemistry and Molecular Biology at Penn State. He is the co-founder of the Center for RNA Molecular Biology at Penn State. There is even a lab named after him on Penn State's campus called the Bevilacqua Lab. And more importantly, they retweeted tonight's event. So Phil, thank you for helping promote this event. I saw that show up on social media, so thank you. He is the author of over 200 publications, and his research has been recognized by the National Science Foundation, the American Association for the Advancement of Science. Now, while he obtained his Ph.D. in chemistry from the University of Rochester in 1993, we are most proud of the bachelor's degree he got at John Carroll in 1987. So I'm going to hand it over to Mike Martin, who's going to moderate our conversation. Dr. Martin.
Great. Thank you, Roy. Welcome to everybody tonight, but of course, welcome to Phil, our guest this evening. So Phil, let's get right to it. Can you just tell us a bit about yourself and how you found your way to John Carroll?
Thanks, Mike. And thanks, Rory, for that very kind and moving introduction. Right. So I was born in Western New York, so not too far from John Carroll. I went to a public high school there. Give a shout out to North Collins High School, and I was interested in the features of a Jesuit education, and I learned about John Carroll and ended up in John Carroll University. As Rory said, I was a graduate in 1987, and then I went on to get my Ph.D. At Rochester, University of Rochester, and then I went in and would postdoc out west at University of Colorado in Boulder. And let's see, I was there from 93 to 97. And then from there, I started my independent career at Penn State in the chemistry department. And I've been there ever since. And so I think that's 27 years that I've been at Penn State.
Who did you work with at Boulder?
Yeah, so I worked with Tom Cech, and he's a Nobel laureate in chemistry, and he's recognized for discovering that RNA could be an enzyme, and coined the term ribozymes.
Phil, I'm going to guess that you're the only John Carroll graduate who's collaborated with 2 Nobel Prize winners. I'm fairly, I can't say for sure, but I think it's a good guess at least.
Yeah, it's actually 3, Mike,
yeah.
but that's okay.
Yeah.
So the 2 collaborations, right? So Katie, who we're here to talk, Katie Carrico and Jonathan Weissman, but then also with Tom, and then also back then Jennifer Doudna was in the lab then, and I think I've one paper with Tom and Jennifer together. So yeah, so it's been really special to have that.
That's great. Phil, looking back on your time at John Carroll and your John Carroll education, what do you consider to be some of the most important aspects of that time?
Yeah, boy, it's hard to answer that because it was such an important time and informative time. The people I met, my roommates, my friends, they were really the values that they had, and also both the education that we took very seriously and the fun that we had. The faculty were super important, and there are 2 in particular that were really influential to me. One was a professor named Nick Baumgardner, who I just heard is still involved with Carroll. So shout out to Dr. Baumgardner. When I started, I was thinking about pre-med and a biology major, and I fell in love with chemistry, and I learned that he had been, I think, a triple major in chemistry, math, and physics. And so I got interested in that. I ended up with my degree in chemistry and a minor in physics. And then I got drawn into biology. There was a professor Robert Bohinski, who had written his own textbook in biochemistry. And I sat in my second semester as a senior, and I took his course, his intro course in biochemistry, and I thought it was super interesting. And I remember him drawing a curve one day on the chalkboard. Maybe it was the first day of class, and it was one of these exponentially increasing curves. And he said, "We're here right now," and it was sort of in the start of the exponential. And he said, "This is where we're going to go." And I didn't know enough to know that that was true or not true, but I thought, well, I want to be on it. I want to be in an area that's like that. And he was a very inspirational teacher as well. And so from there, that to me, that was sort of the seed that really got me doing that. But also really important was kind of the liberal arts education and the rigor, and both in thinking and in writing. We had to do so much writing. We took 3 courses in philosophy, 2 courses in theology, and as I moved along, I really enjoyed literature, and so I took a lot of English literature, American literature. I remember when I was, a second semester senior, I took a course in Russian literature from this professor. I remembered his name when I was reflecting for this interview, and his name is Arthur Trace. And it turns out so I Googled him, and it turns out he's quite a famous person. There's a whole Wikipedia page on Arthur Trace and his contributions to Dostoevsky in particular. So yeah, I would say it was the whole combination of all those things, Mike, that were really important.
Yeah, it's great, to have those moments that kind of shape your trajectory and point you in different directions, and to think about the sciences not as silos, but to get in there and dig in between the departments is a great thing. What really drew you to a career in research?
Yeah, I would say it was it was a little slow for me for that to happen. I started graduate school, and getting into research was challenging. It was so different from what I had done before, from the book knowledge that I had, to sort of moving towards learning what was already known, to creating your own knowledge. I remember my PhD advisor, a guy named Doug Turner, super. In fact, both my PhD and my postdoc advisor, we already talked about Tom Chuck, both super influential on me as a scientist and as a person. And they're both still important people in my lives. They were young when I was in their groups. And Doug used to say that knowledge is sort of like a tunnel. It's not too bad to get to the end of the tunnel, but it's really hard to make the tunnel longer. And and in research, that's what we're trying to do. And it's challenging. But there's sort of many answers to that. I think one is that we can contribute information and new knowledge that has the potential to change the world. So, for example, the mRNA vaccines are an example of that were the beginning of the end, or the way to control the COVID-19 pandemic. But for me, there's really more sort of fundamental and basic reasons. One is to help other people. I really enjoy the interactions with my research group. I've trained over my independent career just under 30 PhDs and over 100 undergraduates. And those relationships and the building of their own careers is really deeply meaningful. But I would say that one of the primary driving forces for me is it's just my own curiosity. It's a way to really satisfy my own curiosity about the natural world. I really look at myself as kind of a seeker, and I'm trying to always create new knowledge and teach it and share it. So it's really kind of a combination of all those things.
Great. So, your primary area of research is what we consider to be basic science, as opposed to some clinical research that might be, dealing with medicines and taking things out into
Right.
kind of human health. Why do you find great value in the government investing in basic research?
Yeah, it's a great question, and and the research that we do is funded by largely funded by the government, funded by the National Institutes of Health, NIH, the National Science Foundation, the NSF, and other institutions. And the listeners to this program, the taxpayers, are paying for that, right? And so that's always a good question. What comes from basic research? And I think we'll see in the work that I'll describe today, that when we started doing this, it was curiosity driven. It was trying to understand the basic functions of RNA and so on. But from that led to important developments that helped contribute to the mRNA vaccines. But other simple examples over history would be the discovery of penicillin, for example, and other cases where basic research leads to aha moments, and you realize that by connecting some of these ideas that you can translate that into really important breakthroughs for humanity.
So I believe you have a slide deck that you prepared to explain some of the background information related to your research interests, Phil. I'll let you kind of take over here, and you can go ahead and start the education portion.
Yeah, I think I have about 10 slides or so, just to give everybody an idea of what this RNA molecule is and how we think about it. And it'll just take a few minutes. Let me go ahead and pull that up.
Sure.
And share my screen. Let's see. Okay. So you should see a slide that says overview of RNA.
We see it, Phil.
Okay, great. So yeah, so I'll just try to give everyone kind of an overview of how we think about RNA. On the left here is kind of a very simple RNA. It almost looks like DNA. You recognize the two strands in the right-handed double helix. And then here is kind of a more globular view of RNA. And then this strange, why I have a picture of a Picasso painting here will, I think I'll make this clear in just a second. So even for those out there who aren't chemists, and when they hear about chemistry, they say, oh, it brings back bad memories of my, high school or college course. I'm going to give hopefully that won't be the case here. I'll give just a little bit of background to some of the chemistry. And so basically in biology, which is made up, of course, of molecules, we can kind of think of them as two types, small ones and big ones. And the small molecules are typically referred to as metabolites. They don't have any repeating structural units. Most drugs that are marketed are small molecules. On the other hand, we have macromolecules. They're very large, and they typically have repeating structural units. That's what RNA is going to be. It's going to be a macromolecule. Here's some examples of small molecules. You can see they look relatively small. We have sugars and vitamins and cholesterol. Over here, we have polymers that repeat over and over again. And familiar ones like DNA, the right-handed double helix, we can see the two strands interacting with one another. And then we have proteins here. You can see different features like this coil and these strands that make up sheets and secondary structures that are common to proteins. And then our favorite molecule, of course, we have RNA. And we can see that RNA, it looks a little bit like a DNA, and it looks a little bit like a protein, right? It kind of has both. So we can see the helical elements. We can see the backbone I've colored the same way because it's a sugar phosphate backbone. So in some ways it's DNA-like, can hold genetic information, but it's also protein-like. You can see it's kind of more globular and bigger in its shape, and so it can fold like a protein. And so these polymers like RNA, we can think of them quite simply as monomers connected together, and I've just colored them, but each one would be, a different chemical structure. So we can have a green one and an orange one and so on. And then the green one can repeat, and they can repeat in any order, and we call that the sequence. And there's a directionality to this. The left is different from the right. The left is the 5' end and the right is the 3' end. And so that's RNA is one of these polymers. Here's an actual picture of our favorite molecule, the RNA, and here's the monomer. And the monomer has 3 units. It has a phosphate and a sugar that makes the backbone, the sugar phosphate backbone. And there's the side chain that will vary with each monomer in our macromolecule. And there's 4 of these side chains. I'm just showing simply 3 of them here. And we give them abbreviations A, G, C, and U. And again, they can go in any sequence. And as I said, we tend to think of RNA structure in some ways as being more protein-like than DNA-like, even though the two are more similar in chemical makeup. And again, we can see here, I've made it most obvious, this particular piece of RNA. You know, it's almost like a roller coaster that you would see where you go up and you try to follow the strands, and it goes around and it comes up and it makes a big loop and it comes down and goes around. And the topology is fascinatingly complex. And so because of this much greater molecular complexity in RNA, we have much greater functional complexity, and RNA can do many things that DNA cannot do. So here's those of you may recall back to your high school biology, the so-called central dogma of molecular biology, the flow of information. And we have DNA here, and DNA holds all the genetic information. And during replication, we get copies of DNA. And then over here, we have proteins, which have a lot of the function. And for a long time, RNA was kind of the poor kind of middleman, didn't get a whole lot of respect in the whole deal, because on the left, we have genetic information, on the right, we have function. But we've come to realize over the last, years, decades, that RNA actually regulates all these steps. It regulates how DNA turns into RNA through making transcription. It regulates how RNA gets turned into proteins through translation. I might come back to that a little bit later. And all these steps are regulated by RNA. And so the view that we tend to take now is that RNA is the center of everything. And it has both properties of both of those molecules. And just to kind of give our audience a sense of what RNA is, it gains its properties by something we call folding. And so I have a little movie here, and in this movie, what you're going to see is this strand of RNA fold up on itself. And so right now it's kind of like a cooked piece of spaghetti, right? It doesn't have any structure, and you kind of stretch it out. But what we're going to see is segments come together through this base pairing. We're going to see them, and the colors are going to match. We're going to see the red pair with the red and a long distance pairing, and we'll see a shorter pairing of the purple and the purple and the yellow and the yellow. And so we'll just let this go. And so as it goes, we see it assemble, see the colors. There's the green and the green. Here comes the red and the yellow, and it assumes this 2D structure. And then other interactions lead to this 3D structure. And it's pretty amazing, right? So that piece of spaghetti is now an L-shaped molecule. And of course, those who have studied some molecular biology might recognize this as tRNA, or transfer RNA. And here's some other views of tRNA. Again, you can see the L shape through the folding and those interactions. And function follows form, and we can see where the different features of the RNA are. So down here is anticodon, where we would pair with the mRNA that was part of the vaccine. And up here, we accept the amino acid, which will grow into the growing polypeptide chain. And it all comes because of that. But what's really important as a background here is that RNA, it has the interactions of DNA, but then so much more on top of it. So this is a perfect example. So here's an interaction that's in the center. It's the one that's kind of in the balls, and you can see it's made out of 3 bases interacting. So we have the regular Watson-Crick DNA base pair, and then a third one comes in and forms a triple. a base triple. And so that's special. You don't see that in DNA. And there are all kinds of these non-Watson-Crick interactions. So there's the one I just showed you with 3 bases and a triple. We can have G not pairing with C. It can base pair with U. That's unusual. Or it can base pair with C in a different way. So instead of having these 3 sticks or 3 hydrogen bonds, it can have only 2. Or we can have bases base pair in even more strange ways than G with U. We can have a big one with a big base and a GA. And so the way I tend to think about RNA is sort of like a Picasso painting. And I had read this quote from Picasso, and that is, everything you can imagine is real. And that's how we think about RNA. If we can imagine it, if it doesn't defy the rules of chemistry, then it's likely to happen, and indeed does. So the RNA structure can get super complicated. Don't worry, I'm not going to take you through this, but this is a picture of part of the ribosome. It was part of a Nobel Prize, and I think it was in 2000. It was around in 2000, maybe it was a little bit after, for the structure of the ribosomes. And this is just half of it, but it's super complex, and you can see the different pairings and unpaired regions. And to me, I often think of this as a little bit like spending a lot of time in airports, a little bit like the Philly airport, with all the different extensions coming off of it in terms of its complexity. But what I wanted to do was just show you, because we want to talk about the Nobel Prize in this year in medicine, and we're going to need to know about the modifications of the bases. And so we're going to start here just with a little bit more technical, but not too technical. So so the 4 bases that are normally in RNA, there's 2 well, let's call them big ones, okay? So they have 2 cyclical features, right? As you can see, a 6-membered 1 through 6 and a 5-member, and they're fused together. And we call those purines, so G and A. And we have 2 small ones in RNA. There's C and U, and these have only 1 ring. And they have names, purine and pyrimidines. We won't worry about them. Let's just say we have 2 big ones and 2 little ones. There's only 4 side chains, and they look pretty similar, right? I mean, these 2 don't look too different from one another, and these 2 don't look too different. They have the same atoms, more or less. So how much can we do with that? Maybe not as much as we would like. Proteins have 20 amino acids, but one of the ways that RNA gains diversification is through modifications. And so this was really important in the Nobel Prize. So if I go back to this slide, here's U, one of the 4 bases. But there's modifications of U, and they're subtle. When I made this slide, I color-coded the atoms so we could sort of see. Here's U, and the variation is called pseudo-U because it's U-like, but it's not U. And so what you can see, the easiest way to give it away is where you can see the base is attached to the sugar. It's attached through in normal U by a nitrogen carbon bond, but that's gone. That nitrogen, that aqua or teal nitrogen, is moved over here in the ring position, and there's now a carbon-carbon bond. And that was really key in the mRNA vaccines. Our RNA, human RNA, is filled with these modifications. Pseudo-U is one of them. There's another one that was really important, in which this hydrogen is replaced with a methyl, and that's 1-methyl pseudo-U. Again, you have the carbon-carbon bond. And so one of the big breakthroughs was the incorporation of these non-native so-called modified bases. So Mike, I'm gonna escape out of here and bring it back to continuing our conversation.
Great, thanks, Phil. I think about this, this RNA modification, right? So, a U is Phil. Phil comes home, family recognizes him, right? The pseudo U is like Phil, has a mustache, right? It looks a little different, right? Not quite the same, but pretty much the same. I mean, it's a little tougher to recognize, maybe, but pseudo U, pseudo Phil, right? And then an even different type of modification. Maybe the methyl group is added. Right. Phil's, you've got to get a mustache and a wig on it, right? It's a little maybe even more difficult to recognize. You don't look the same. You're kind of the same, but not quite the same. Like, how does the cell, kind of differentiate and kind of understand that, and how does it affect the cell's ability to work, really?
Yeah, and biology does this repeatedly. You see it not just in humans, but it happens in bacteria and in other systems as well. And so one way to distinguish what we call self and non-self, how do I know it's me versus how do I know that there's not an infection by a virus? And so one way to do that is to mark your RNA with a mustache, if you will, with the pseudouridine. And so I can say, aha, this is normal cellular RNA. Okay, no need to get alarmed here, versus something else comes in that doesn't have it, or it folds differently. So I showed the fold of the L-shaped RNA, but another RNA comes in that folds. In particular, the fold is a kind of a boring DNA-like fold where it's just an extended RNA with lots of base pairs. We don't see that in human RNA. And what we think the reason is that's a way, both by the shapes of the folds and the chemical makeup with the pseudo-U's, the mustaches, those are ways to tell it's self versus non-self. So it's not only, part of it is how cellular RNA behaves, and then there's also marks that give clues that another RNA comes from a virus. Not only the absence of pseudo-U, but the presence of other chemical modifications. How I showed that the RNA has an end, that the 5' end has a certain modification. So the viral one, might come in with a certain hat, a baseball hat on the end, and ours is, not a baseball hat, but kind of like a cowboy hat or something like that. And so those signatures also lead to marks that we call PAMPs, PAMP, for pathogen-associated molecular patterns. That's a mouthful. But basically, at the chemical level, what is it that tells us that this is the bad guy that's not supposed to be there?
Yeah, we have to be really efficient at recognizing, non-self, and really good at not recognizing self, right? To prevent
Yeah,
things like autoimmune diseases and whatnot.
right.
A very complicated system that's based on very kind of simple things, right?
That's right.
Short sequences of proteins and modifications to RNA. Um, what do you find is like the real practical purpose then of your research, your lab?
Yeah, I mean, extending out from what the funny thing about the mRNA vaccines is that we were really curiosity driven, looking at this process of immunity. Most of our audience probably is familiar with acquired immunity. For example, if you get a booster shot or you're exposed to a vaccine that's not live, so that's sort of let's say for mumps or measles or something like that, and you acquire lifelong immunity. That's not the immunity that we're talking about. That's a special immunity called acquired immunity, which is fascinating. But the one that's important for the mRNA vaccine is really innate immunity, and it's something that's thought to be ancient and recognizes these molecular patterns. So when we started working on this, and this is in the first half of my career, we really worked that was earlier than 2010. I would say about 2014 or so, we switched and began working on other things. But at the time when we were working on this, we were really just trying to understand how RNA could be distinguished, as we said, between self and non-self. We weren't trying to create a new vaccine or anything like that. In fact, one day what happened was that Katie Carrico at UPenn. So Penn State is Pennsylvania is kind of like a rectangle. Penn State is smack dab in the center, and UPenn is over here down in the corner at Philly, and contacted my lab. They were aware of some of the work we had done on some of these modified bases and suppression of this immune response or this inflammation response on a particular sensor called PKR. And she said she was working on something that she called an mRNA vaccine. Well, I'd never heard of an mRNA vaccine, and I'd heard of mRNA, and I'd heard of a vaccine. And she said she'd been working on this her whole life, and they wanted to do some work together to test some ideas. And I said, all right, you seem really like you've got some really great ideas, and I'll try anything once, so let's try it. And we started working together on that. She sent a student up, and that's the first time we really looked at mRNAs. She had really been working on that her whole life. And that's kind of how it went, and I would say, and then we incorporated pseudouridine into the transcripts, and we saw a very, very different result in the fact that under those cases, it was a suppression of the immune response, and so protein production was really nice. It didn't get suppressed or anything like that. And quite honestly, like I said, 3 or 4 years later, our group had moved on to other things that we do now. We got really involved, and we still work heavily in plant biology. We began to develop techniques to look at RNA structure genome-wide. We really moved into other directions that I don't have time to talk about. And then around 2020, the global pandemic struck, right? And just like that, and we began to hear about the need for a vaccine. And both Katie and Drew and other people I knew, Moderna and Pfizer and so on, Pfizer and biotech, just basically in record unbelievable time, right? They were able to design these RNAs in a very short time, a matter of weeks. Once they had the sequence of the virus, and then we were delighted to see that happened, right? So yeah, that's kind of a little bit how it went.
Yeah, I think you make some good points there, Phil. I think the arc of scientific research really tells a story, and there's a number of chapters that come out intermittently. And each chapter is represented by some publication, and maybe you're co-authoring, so you're writing together with other people, right? And I just want to take a moment so the audience kind of gets to clue in once again on that timeline, is that in 2010 you're publishing a paper in nucleic acids research that's based, that's a follow-up on research that you had done previously, at least as early as 2008, but probably the research is pushing even further back than that.
Yeah.
And then there's a whole decade before it hits the scene in ways that people, and including you, may not have predicted is going to happen, right? Or maybe be needed in our own lifetime, right? So and these stories are told by people, right? So how many people do you think, undergraduates, graduate students, postdocs, collaborators, help to tell this story?
Yeah, I mean, I can definitely talk about that. We'll say one thing that's really interesting. We'll come to that in 1 second, but you talked about the time lag. You could go onto Google Scholar and look this paper up and look at how many times it's been referenced. And now it's been referenced over 500 times, but it didn't get referenced for about 10 years or something like that. The references, it's kind of like that exponential, but really long incubation phase before the exponential happens. Nothing, everybody kind of forgot about it. I forgot about it, to be perfectly honest with you, because we all move on to other things. But I think what's amazing, particularly, I would encourage the audience to pick up the book, which I'm in the middle of right now, on the autobiography of Katie Carrico, which is fascinating because she was not a tenure track professor like I am with the privileges that I have, but it was really more, she was a research professor, right? And there went through a ton of struggles, but really stuck with this idea for a super long time. And the number of people that were involved with this in my own lab, it's not a huge number, but probably 10 or so people over the years. There were some key graduate students and postdocs and a few undergraduates that all were involved and worked on this, but also didn't realize at the time, had no idea. I had no idea that this would, kind of happen. So
Yeah, I mean, that really gets at the power of basic research. We do
right,
the work to generate some knowledge, and then, who knows where it goes in 5,
right.
10, 20 years. So before we move on to Q&A, Phil, I just
Yeah,
want to give you a moment to maybe wrap up with some, specific reflections you might have on, John Carroll, or in your education there, or any Jesuit education you might have.
sure. Let's see. You know, I think for me, one of the things that was key for me in in any success that I had over the years is really the curiosity to learn new things and to strive to be, a creative person. I think that's a fundamental thing. It's a core thing, and I think that the people I met and the people I interacted with, the professors, the priests, the students, all those things were just really key. And I would also say that another skill that I developed there was the ability to write well. And it's surprising how much time, surprising to not to me anymore, but to a lot of people, how much time we spend writing it, whether it's an email or a grant proposal or a research paper. And I learned at John Carroll how to do that. Even when I didn't know I was learning how to do that, right? And
Yeah,
so I think that was really key.
all right, great. Good stuff there. I try to remind my students that they're creative people. Their creativity may not look like a painting necessarily, but to be creative in science may be to design that experiment. Right?
Uh,
So
And to
uh,
stick with it, it takes time,
yeah, yeah, the instant gratification is not always there in science, but um, I don't know if this will make you feel good or bad. Or students only have to take 2 philosophy courses now, so not as many as they used to take back then. But
Now, the
uh,
hardest courses I ever took were philosophy
yeah,
courses. They were just, for me at least, the way my brain works, it was very challenging to take those courses.
yeah.
Yeah,
Hi, Phil. So we have some questions from the audience here. Have you heard of the Code Breaker by an author, Walter Jacobson?
I've heard of it. I haven't read it. Yeah, I probably
Okay.
should, but is there a,
That was the question.
okay.
No, no,
No, I'm gonna write it down. I got a Post-it note right here on my desk, and I will, uh, oh wait, wait, the Code Breaker, is this the one about Jennifer Doudna? I'm always getting the title. I think it is. Hold on one second. Now we're really doing this live.
I see. Maybe it is yes, but
It
maybe
is. Oh yeah,
it's
yeah. I
different
read
for
this
you.
book.
Okay.
Sorry, I didn't remember the title. Sure, I read the book. Yes, and it's a great book.
Yeah.
And some of the things that happened in the early parts on with Jennifer. Sorry, yeah. That was when I was in the lab, so I was actually part of that, right? So we overlapped. I was in Boulder from I always get my years correct, from 93 to 97. And I think that Jen was there from 90, we overlapped for 2 years. I think she was there a year before me and was there until about 95 or so. So yeah, I got to see a lot of those things and got to see some really exciting things happen at the time. You know, when I was in the lab, and this is only a little bit in the book because it focuses mostly on CRISPR and gene editing, but was they worked out the second structure ever on an RNA, and that happened when I was in the lab. And she was the lead author on that in the Czech lab. And that was a piece of the group one intron. That was the catalytic RNA. Of course, the first one was tRNA, the L-shaped one that I showed. tRNA is pretty the first obvious one to do because you can isolate it from the cell, but all the other ones were much more difficult. And so it took it took 20 years for that one to come out, for the next one to come out, a long time. And that opened the floodgates after they solved that structure. And now we have hundreds and hundreds of RNA structures. So super exciting time.
I saw there was a few of my current students who are attending, and you showed multiple different ways that bases can base pair with one another. You certainly ruined my genetics questions. I have to accept all answers now for what pairs with this sequence. I can't say as I'm happy about that, but this next question may have come from one of these students. So what advice would you give to students and young graduates who are interested in a career in research and teaching?
Yeah, I mean, I think you should find things that that you love. I think that's key. You have to have an enjoyment. The people I know who have been really excelled, right? These Nobel laureates and things like this. They are, when you talk to them, they're just fascinated by what they do. And so find something that is both interesting to you and then approach it in a way that you sort of have a natural gift for doing. I remember, my Ph.D. advisor, he said, well, you're good, Phil, you're good at doing mathematical problems. And, it's great that you're in this area, in this mechanistic biological stuff, but like, that's like your key advantage. And he was right. It didn't matter. There are lots of problems that you can take on that are very important, whether it's gene editing or mRNA vaccines or something like that. But you want to bring something new so that it's recognizable that it comes from you. So and it's hard to say what that is. People love different things for different reasons, some of which you can't put your finger on it, right? So one person loves to listen to classical music, and another person loves jazz, and another person likes popular music, right? And so it's hard to explain why. You just do sometimes, and it'll be the same thing with you try to appreciate it all, but it's really to just kind of follow your gut sometimes.
Right. So how does the structure of mRNA allow for wobble base pairing?
We don't know much about the structure of mRNA. It's a really, really good question. So I showed some structures in there, and we're working on that. I showed some structures of highly structured RNAs. I showed the L-shaped tRNA with the base triples and the weird, upside down base pairs and the two big bases and all that kind of stuff. And then I showed the ribosomal RNA that looked like the Philadelphia airport, right? But neither of those were messenger RNAs. Those were ribosomal RNAs and tRNAs. The messenger RNAs, which carry the information to make the proteins, which were the mRNA vaccines, we don't know much about how they fold. And we don't think they're all that structured. We don't think that they're these globular entities. And so, in fact, our lab works on that right now. With my colleague Sally Osman, who is in the biology department, a plant biologist, we invented a method about a decade ago to look at this, to begin to get information about the structures how do you get that information? And so I won't get too technical here, but basically, when a molecule folds, then there's regions that are structured and they're protected from chemicals, and there's regions that are loopy and those unpaired regions are exposed. And we can figure out what those are by chemicals that react with the exposed and not the other ones, and using different molecular biology techniques to get that out. But I will say, though, that the mRNA vaccines, there's a drive to make them as structured as possible. mRNA, and I highly recommend that. As good as the code breaker is, whoever asked me about the code breaker, okay, I don't want to disparage the code breaker. It's a great book. But you got to read Kariko's book, because the code breaker is a biography about Doudna, and the book from Kariko is an autobiography, and it really talks about the efforts and the passion in doing all of these different things. So.
All right, so what other career opportunities did you happen to consider before arriving at Penn State?
Which is called Breaking Through. The book from Kariko is called Breaking Through. What other career options? I never considered another career option. This was my only career. I 100% wanted to do this. I mean, I was lucky because I never struggled like, oh, do I want to go to graduate school? I knew I wanted to go to graduate school. And once I did that, I knew I wanted to postdoc for Tom. And after that, I knew I wanted to go into academia. So for me, there was never another choice. I was sort of, I guess, fortunate in that way. If I had to choose another thing, I mean, I love to teach so much. I think I still probably would have been a faculty member. I love literature, and I love talking about writing and poetry, and I love to write. So maybe I would have done that. But there weren't a whole lot of other things that I considered. I was all in, I guess.
Yeah. Great. I guess one more coming in here. Could you describe the techniques you used to study mRNA in your lab?
Sure. I started to talk about one a little bit, and it's really so there's two ways to study RNA. You can study it in the test tube, which we call in vitro, and then we can try to study it in a living organism, which we call in vivo. And you might guess correctly that it would be more relevant to study it in vivo. And so most of what we do is try to study it in cells, and we collaborate with because we're chemists, really. We collaborate with biologists. We've applied that technique that I mentioned about probing the structure of RNA, and we started in model plant systems called Arabidopsis, which is like it's not an important crop or anything. I guess you could eat it, but it's kind of a bitter mustard green. And I started to apply it to crops, particularly in rice. And then we've looked in bacteria, which of course serve two roles. They can be really beneficial in the microbiome, but they can also be really toxic in other cases, in archaea, in vaccines, and so on. So the techniques that we apply there are really broad. So we're doing chemical synthesis. For those who are listening and might be chemists, making molecules that can go in through the cell wall and through the membrane, react with the RNAs. So there's chemistry, new reagents to attack new parts of the bases and new faces and the sugar phosphate. But then there's molecular biology. Once we modify the RNA, how do we get that information out? And using specialized enzymes that turn the RNA into DNA and read through the modifications. Then after that, there's genomics. We have to make libraries and sequence all of this. And then after that, there's computer science. It's really big data. Sometimes we're getting out hundreds of millions of reads, or even billions. And so like terabytes of data. And so someone's got to analyze all of this and make sense out of it. So we bring together teams. My research group is quite diverse in terms of, we've got chemists, biochemists and biologists and computer scientists, and so it stretches me, and that makes it it's good. It's good and bad. It's mostly good, but it's challenging, and so it's really a combination. It's what we call interdisciplinary science. Those are the techniques that we've developed to take on some of those problems.
All right, thanks, Phil. I'm going to throw this down to Rory here.
Well, thank you, Mike, and thank you, Phil. That was a wonderful program. Thank you for taking the time to be with us this evening. Thank you all for your questions, for those for tuning in. Just a couple of notes here. Again, thank you to both our moderator and our presenter. Thank you for everyone coming in. One more plug for John Carroll. For those who are unaware, the John Carroll men's basketball team is in the first round of the NCAA tournament. We're hosting a game this Friday on campus for those Clevelanders in Northeast Ohio. The game's at 8:20. We play SUNY New Paltz in the DeCarlo Varsity Center. We're having a reception beforehand in the renovated DJ Lombardo Student Center. If our athletic director was on here, he'd be hitting me over the head, making sure I promoted this. So I want to make sure I do that because there's a lot of excitement on campus for the team and for the community. So that concludes our program this evening. I hope you all enjoyed your time. Thank you so much for engaging with John Carroll, and thank you again to Phil and Mike for this evening's program. All right.
Thank you.
Good night, everybody.